But it is quite tricky because there's a number of different factors that are involved, going from the very fine detail of molecular biochemical pathways into the whole plant, different canopy processes, and then with sugarcane as well, we have the multi-crops cycle causing complexity as well, going from plant to ratoon crops. So all these factors can be important at different times and also interact with the different environmental conditions as well. So that makes it quite a challenging task to implement this sort of screening into breeding programs. But the one thing we're trying to do as well is look at how the different genotypes or cane varieties interact in the soil process. So here we've got a schematic of how nitrogen changes in the soil. So in the soil there's a very large organic nitrogen pool or protein, and that breaks down. When we put urea into the system, have a much larger increase in one of the forms ammonium, and then that moves on to nitrate, and that's the one that's really easily lost from the system. So a lot of our projects that you're involved in and others are targeting trying to reduce these nitrification rates and look at different ways that we can do that. The other way that we can also look at it is trying to increase the crop's ability to use nitrate. So that's one of the areas that we've been looking at as well, and how we can get different varieties to take up more nitrate that's around. So that looks back into starting to look at the interactions between plant and different nutrient sources. And so for many years it was sort of thought that just nitrate and ammonium were the only sources of nitrogen that plants used, and nitrate was really the major source because that's what everyone could measure quite readily in the soil. However, after we've been looking at comparing this across different plant species for a while, it became apparent that this wasn't always so straightforward. And we've been comparing in sugarcane how they access nitrate versus ammonium, and then also can they take up the other organic sources straight from the soil. So this has been an area of research for us as well. And some of the work that we found is that when we looked at very small sugarcane crops and offered them the ammonium and the nitrate together, so this is often what you'd see early in season after application of fertilizer, and there in the blue bars you can see that's the ammonium that's taken up as compared to the nitrate. So the sugarcane was taking up a lot more ammonium than it was nitrate, so leaving it there vulnerable to loss. And when we compared this across all other different types of species, so at the top there we could see the commercial sugarcane variety. It had the worst use of nitrate compared to ammonium, moving through the different ancestors of cane, spontaneum and officinarum, and then moving up towards Erianthus, which is a grass that's very closely related and often is bred into the sugarcane varieties for drought resistance and other things in a research capacity. You see that it was able to use nitrate much better than sugarcane. So this was indicating that there was potential for us to maybe increase the nitrate uptake of sugarcane compared to others. And when we tried to understand this a bit more across the different types of crops, we did see when we compared maize and sorghum and Erianthus, they had a lot more nitrate in their tissues compared to the Q cane, which is the one at the end there. So we've gone back, and as part of our trials now, where we have 40 genotypes that we're looking at in a variety trial under different rates of nitrogen additions in terms of trying to characterise different varieties, we've been looking at the variation in nitrate accumulation to see if we can find some varieties that are much better at sucking up and keeping nitrate in the tissues. And so this is work that is ongoing there, looking at it from a plant perspective. And also, as well as that, there are closely related grasses, such as the brachiaria, which have been found to influence the soil nitrogen cycle processes. So these ones are quite closely related to the Erianthus as well, so there's in the same family tree, quite close to the sugarcane and Erianthus. And they produce exudates from their roots, which they pump out into the soil, and it stops that conversion of ammonium to nitrate. So there is some capacity of plants, as well as their ability to take up and use different forms to change the soil nitrogen cycle processes. In a way similar to some of the products that you can buy that are applied, there is potential to use this in some sort of crop rotation where you could have plant crops influencing the nitrogen cycling processes of the soil. And this is one area that is being explored in Brazil by some colleagues where they're both extracting exudates from plants and applying them to try and inhibit things, but then also using rotations of particular plants that have been observed to inhibit nitrification as well as part of their cycle. But this is fairly new work that's going on. So there I've been talking about the different plant and cropping systems and how they might start impacting on the soil from their plant exudates. You can see here, this is where we started to move away from the very plant-focused plant uptake approaches into more a systems approach, and how this is all impacting and interacting with the soil and all the microbes that are interacting in the soil. And this is a diagram that we have here showing all the different components of the microbes in soils, different soil organisms, and how that impacts. You can see the arrow there. So here we were talking about the root exudates and how that might impact on the soil cycling of nitrogen through the influence of different bacteria and microbes in there. And then how do we know what's happening, or how could we say this is like a healthy soil? In the black there won't be hard to see, but there are different parameters that can be measured to try and gauge what's going on in the soil. So how much soil carbon there is, what nematodes you have, what's the microbial biomass, are all things that are starting to be measured in different soils under different managements to try and understand these impacts and interactions. So this is the foundation of a lot of the work that's going on in soil biology, because it is known that changing land use to agricultural land, different agricultural managements, selects for particular soil microbial communities. So which bacteria are present, which fungi are present, and all the different macropods and nematodes as well, and how that's influenced by what we do. And this changes quite a lot when compared to native vegetation that might be nearby the agricultural land. What's also the microbial populations are a lot lower in the sugarcane cropping than has been observed in native systems or those that are close by or only recently cleared. So if we take a step back from that and have a look at why this might be the case, if you have a look at a natural system where you have a lot of no biomass or very little biomass is regularly removed from the system, there's a lot of recycling and decomposition of the carbon. You have a very high soil organic matter with a lot of deeper root growth, and so integration over a much deeper zone of the soil, as compared to the cropping systems where you have a large amount of biomass taken away. Sometimes there is some litter inputs remaining, but there is a large export of carbon from the system. You have much lower soil organic matter and with larger inputs of NPK in the absence or not combined with different carbon. So this changes the cycling quite a lot and allows for much larger loss pathways from the system. So, as I said before, the reasons behind the different microbial communities and changed carbon-nitrogen cycling in those systems can be because of less diverse plant litter, so different types of the makeup of the carbon, how much lignin, or how difficult it is for the different microbes to eat it. Are they used to large inputs of the same amount of plant tissue? Can change which type of bacteria and fungus are present. Also with tillage, it does impact the fungal hyphal network quite a lot, so that can destroy those connections between the fungus and the plant roots. Again, as I said before, the mineral nitrogen is added without any carbon with it as well, so it's changing what the microbes want to eat and what they expel. And also with the monoculture, if there's no rotations, you have an increased loads of pathogens available in that system. And so while it's becoming, the knowledge is increasing on what's present in terms of soil organisms, but you can see there with the different numbers that in the darker red, this is the number of known species. So we know there exists, probably we don't know exactly how many are there, we don't know how they're impacted by different management, we don't know how variable it is, but also alongside that in the lighter colour, if you can see, I'm not sure, below is the estimated species. So not only do we not know what the known species are doing, there's a huge array of soil organisms that we don't know that occur, and so we don't really understand what's going to happen to them under different managements either. So in terms of the way forward, in terms of research, what's going on in soil and the different microbial interactions is hugely unexplored, but is increasing as the techniques come. So you can see here with the increasing technology available, being able to just observe what's there is now much easier to do. So you might think a lot of these techniques were developed in human medical research, and it turns out soil is a much more difficult medium to extract and understand than the human body. But as the techniques are coming, and also as the price to do this type of work is decreasing, we can sample a lot more places, a lot more times. And that's really what's necessary to be able to understand what's changing when we're doing these different practices and over what time frame as well. So here's an example of how in Australia they're trying to characterise the biomes of the soil present under different types of vegetation and different types of cropping. This is an example of a large conglomerate of scientists, and there you've got the map of Australia where they've sampled soil at many different locations to try and determine what's in it. So just like a stamp collecting exercise, I guess, just to see how different all the different soils are. So here's an example where they're processing the sequencing just from those different environments to see what bacteria are there, with differences. In the red arrow is the sugar, that was taken from sites of ours near Mackay and one in Burdekin. As well, and as the fungi, different groups of fungi that are present as well. So this is very base level information. So this is the sort of species that are there. We can try and figure out what they're doing based on what type of bacteria they are, but we really don't know how much they're doing it and how they change with the different management processes. So there's still a long ways to go there. So one thing that we've tried to do is understand how the function of these bacteria change with different management. And at one of the sites we had set up large areas under low nitrogen supply, only about 20 kilograms per hectare, as compared to the recommended, which was 180 kilograms per hectare. And that was there for one crop cycle, so 3 years, a plant crop and 2 ratoons. And at that time, we sampled the soil and the roots to have a look at what the bacteria were doing. And here we've got the graph showing the different genes or the different controls for the different nitrogen cycling. So here we've got the nitrogen cycle, have urea coming in, changing to ammonium and around to nitrate. And from that, we saw that in the high nitrogen treatment, this was a much more active pathway. So once you're putting urea into the system, you have a much greater pathway for bacteria to change the ammonium to nitrate. So this is sort of speeding up or enhancing that pathway. And similarly with denitrification, so changing the nitrate to either nitrous oxide or N2 is also sped up. One thing that we were hoping to look for was that in the lower nitrogen supply, this would aid nitrogen fixation. So that's where atmospheric nitrogen is converted by the bacteria as a nitrogen supply. And often that's what legumes have an interaction with bacteria to have this process, and often it has been supposed that sugarcane may have the ability to have these nitrogen fixes associated with them, so that you could get a lot of nitrogen supply from the bacteria, not the fertilizer. But unfortunately, in the last section here, we didn't really see any upregulation or increase in the bacteria that would do the nitrogen fixation when there were low levels of applied nitrogen. But this is a fairly complex process, and you'd have to do that probably at a number of other sites and over different time scales to really understand that. And this was very soil-based as well. We didn't look at it in different parts of the sugarcane system either, so that's yet to be explored in more detail. So as well as focusing on the bacteria, we also had a look at how it changed the fungal community. And so we did get a different fungal communities associated with both the roots as well as the bulk soil as well. So when there was this change in available nitrogen, we did have a shift in the different populations of fungi present in the soil in those particular cases. And when you look at this in more detail, across here you can see in the grey as well, there was a fairly large number of fungi that were unidentified, so we don't really know what they're doing or what characteristics they have. But for the ones that were identified, they can be allocated into different types. So some that might have been associated with biocontrol in different situations, ones that were different pathogens, or ones that were decomposers. And what we did find, if you look there for those that are highlighted in red, you can see in the top section here, this was the low nitrogen treatment, and in the high nitrogen treatment, we did get an increase in pathogens of fungi that were classified as pathogens in that particular soil. So you can have different management practices, and here it was just the only difference was the loading of nitrogen that was applied to the system, and also probably this was associated with a yield decrease at low nitrogen as a high, but we did have changed amounts of fungal pathogens in response to higher nitrogen applications in this setting. As well as the bacteria and fungal pathogen populations, there's also been some work done on nematodes and different nematode populations that was done by Graham Sterling at the same sites, and also associated with higher nitrogen applications were increases in the amount of pathogenic nematodes and some decreases in the amount of beneficial nematodes associated with that increased nitrogen application. So this is a snapshot of different bacteria, fungus and other nematodes, and they could have impacts on the yields of those things, trying to understand what the changes in these populations are with just changed nitrogen supply. So this is just the amount of urea going on. There is a whole other implications as well if you have different rotations. So a lot of work has been done looking at legume rotations and intercropping and looking at the different pathogen breaks and other things associated with those farming systems. So the other area, and because the stimulated interest and understanding of the importance of the microbes in this system, a large area of research is focused on looking at beneficial microbes. And so globally, there's a large input, usually in very contained and controlled conditions in the laboratory, that have demonstrated that plant growth can be enhanced by addition of beneficial microbes. And here's an example of some work we've done where you can have no microbes, then when you add microbes, you have an enhanced growth. So this can be due to a number of different factors. Can be a nutrient input, so some bacteria are able to supply different nitrogen, different sources of nutrients from nitrogen. They can help mobilise phosphorus, but also it can be related to plant hormones as well, or things that stimulate growth. So that is one of the main mechanisms of action for these things, and also as pathogen suppressants. However, while this is very well established and the presence of microbes can be seen entering the root here, so being taken up by the plant, and also forming some biofilms around the edge of roots, it's much harder to be able to demonstrate this effectiveness of inoculants in field settings. So it is very difficult to be able to demonstrate yield benefits in field. But if you think about in the laboratory environment, when you're adding microbes in, you can be assured that they're present during the stage of plant growth. Once you go into a field setting, it can be much more difficult to know if the microbe that you've added is still there, how long it's there for, and what sort of influence it might be having in the area that's been applied. So currently there's been more of a focus, I think, on testing the effectiveness of these biostimulants and probiotics in the field. One of which a student in our group, Shelby Berg, has been involved in the Herbert, trialling different products, then testing the amount of microbial inoculant present in the soil at periods of time after addition. And then there's another article there that came out from Susanna and Schmidt and others who I work with, looking at if you want to have a look at crop probiotics in a wider sense, and really the need for a lot more scientific testing in this area. So if you have a look at the area of impact, so it's really the nutrient and rhizosphere that we're looking at. So here in this diagram, we've got a picture of plant roots, and then around that you can have an area of exudation, and this is also on the surface of the root. This is where you'll have all the microbes being attracted to eat the exudates, and also this will be influencing the amount of nitrate and water and phosphorus that's available as well. So you can imagine it is quite difficult to be able to sample this area and understand what's going on at such a fine resolution. And so there will be quite a large difference between the rhizosphere and the bulk soil. So often when we're measuring bulk soil, we might be really missing the processes that we want to understand. So with the techniques and different tools that are becoming available, with different root probes, growing plants in different environments, being able to understand the processes at this interface is really what's required to enhance plant and crop nutrient uptake and enable soil health, I think. So here again we have those factors that are going to influence this. It would be the genetic makeup of the crop, what chemicals are released, when they're released, how that impacts on the soil biological community at all those different levels, from the bacteria right up to the macropods. And then how is overlaid on this is the different environmental conditions as well. When it's raining, what the temperature is, and at what point in the season that this is occurring. So it is a very complex situation. However, what we're looking to do is really synchronise this nitrogen supply and other nutrients as well with the crop demand. So at the moment, when we have a look at the soil availability after addition of fertilizers, you can see in the red zone the nutrients are very available for uptake when there's not a lot of roots colonising that area, or when the plant demand or the plant crop is very small. So it has a very low capacity to store the nutrients as well. So what we really want to do is move to this other area here with next-gen fertilizers, where there is a better matching of the release of the nutrients with the crop demand for nutrients. And this is an area that's been targeted in many different ways, one of which is with the current enhanced efficiency fertilizers, where you have the application or combination of those nitrification inhibitors with the mineral fertilizer. So they're trying to slow that process of nitrate production to allow ammonium maybe to be taken up, and it binds to the soil, it's not as easily lost. And also with plastic-coated formulations that are slowing the release of urea or others and the interaction with the water and the microbes in the soil. And there's a lot of the testing, field testing currently going on associated with these products. But there also has been some moves recently to try and understand what the other impacts of these products are on the environment. And recently there was an article that came out looking at the effect of these microplastics, and microplastics in waterways, but also in soil, are starting to be acknowledged as maybe having potential problems and downstream effects. And so here's a release of a study where they're looking at the effect of the microplastics from fertiliser products on worm populations. So I think it is unknown what the impacts of these might be, but it is increasing interest and awareness of the implications of these things. So we were looking at this from more of the combined plant and soil health point of view, and looking at how we can optimise nutrient supply. So currently in the systems we have fairly inefficient mineral fertilisers. Also associated with that, we have degraded agricultural soils where a lot of the organic carbon or the soil organic matter is lost because of various management and changed inputs. But also alongside this, we have a really rich resource in that there's a large amount of nutrient-rich wastes that are being produced that would be able to potentially deliver nutrients and carbon to agricultural soils. And this is the area of research that we've been moving into more recently, is how to improve soil health, but also access a fairly unexplored resource of wastes that can enter into the agricultural nutrient cycling. However, the one thing about these organic wastes is that their nutrient release is fairly inefficient. So it's not, manure has been used for a long, long time as a nutrient resource. However, the release can be quite slow and it's often very variable. So it's really trying to harness the potential or modify these products to be able to have more consistency in their nutrient supply potential. And so that's an area that we've been exploring more recently. Okay, so the term here has been next generation fertilisers, and these are based on repurposing these wastes to be able to deliver the multiple benefits to the cropping systems. So some examples of the organic materials that I'm talking about could be manures. So this is a fairly well-known resource. You'd all be familiar with mill mud, but there's compost and strovite as well, which is a urine part of recycled nutrient resource. And then alongside this, we have the sorbers for on-demand release for plants. So instead of just putting on the waste and incorporating these in, they can be combined with other things such as clays, biochars, different agricultural residues, as well as engineered starches to be able to have a greater control of the release of nitrogen from these waste products. And then there's also the opportunity to be able to incorporate microbes into this mix as well. So often a lot of the microbial inoculants or products, often liquids in vats, but there might be a better chance of the microbes remaining present or surviving and remaining functional if they were encapsulated or incorporated into one of these products rather than as an immediate liquid application phase. So there's a lot of area there that can be explored with the microbes, fungi, bacteria, worm eggs. The list is really endless of what could be incorporated into these things. And so this whole would be for enhancing the soil fertility and providing nutrient, but as well as that, reducing the nitrogen losses to waterways as well as the atmosphere. So the thing is with different wastes is no waste is perfect in terms of its nutrient composition. So the thing about synthetic fertilizers is that you know what's in it, and you can formulate it and give the crop what you want. So the NPK ratio is very well defined. You can have micronutrients and others. However, if you're looking at chicken manure or mill mud or compost, they have quite large amounts of carbon and much smaller amounts of the other elements, and these aren't in the ratios that are required by the crop. So there'll need to be some amendments there with perhaps adding some mineral fertilizers or combining different waste to provide the perfect combination of nutrients for different plants. The other thing is that the nutrient-rich wastes need to be formulated so we can avoid the different losses. So if you applied the manure and compost, and here's an example of a sugarcane field where we've measured the greenhouse gas emissions after applications of different products. So when you're putting a lot of carbon into the system with nitrogen, it's stimulating the microbial activity quite a lot. And this could stimulate it so much that a lot of the nitrogen that you're putting on is going off as greenhouse gases instead of remaining in the soil. So this was an example where they looked at additions of manure and compost combined. So this produced the greatest amount of greenhouse gases compared with just standard urea fertilizer. And then moving down, just manure alone, and then manure combined with some of these sorbers, such as clay and biochar. So when you combine the manure with the clay, it protects or attaches the ammonium that stays in the soil, and then this is able to be released later for the plant to use rather than it rapidly being converted to greenhouse gases. So there is a need to be able to combine these waste products with different sorbers or other elements to control their release. So we've been exploring this work more, trying to see what other materials are there available that can be combined with wastes. And here's an example of some other work we did in the lab where we had the control, which was a combination of sugarcane meal mud and manure. And then we combined that with a whole lot of these different sorbers. So there's different clay minerals that can be mined. And here, we found that the availability of nitrogen in the controls, which was just the waste, the meal mud and the manure, was much greater than the plant would be able to take up at that point in time. But then as you move down from the grey down to the yellow, right down to the red, this particular mineral, clinoptonite, is able to bind the ammonium more tightly than these other ones, and so it had the lowest diffusive flux. So there, it's a much steadier, slower availability of release of ammonium. It's much better to the root's capacity to take it up. So even amongst the different minerals that you can mix in with these different waste, we have different potentials for binding. So you really need to understand what is the best combination of materials to provide the release that is best matched for the plant uptake. So there's a lot of scope and a lot of things to be looked at of how we can manipulate release of nutrients in line with plant uptake at that root interface, and then scaling up to the whole crop system. So as well as looking at the release rate, we also can look at how they interact with plant roots. So often when you have some potential of organic matter or also availability of nutrient, you have some interaction and stimulation of root growth. So a lot of these wastes have been pelletized. So there we form them into pellets and then provide those to plants in controlled systems to see how they interact with plant roots and to see if there's a stimulation of root growth or an enhancement of root growth around these particular products. So a lot of these next generation fertilizers, they will be formulated based on these repurposed wastes that will be able to improve soil health and also use smart biomaterials. So in our latest project, we have been working with chemical engineers to look at different ways to come up with these different matrices where we have different combinations of materials that are able to have a more controlled release. And so here we have examples such as this where you have clay that is bound with different urea complexes and then immersed into a biodegradable polymer that has different release characteristics. So this is quite a layered, complex approach of different combinations of urea, of different wastes, also including different starches that could be available from different agricultural residues, which all combine together to form a matrix which allows the nutrients just to be released in response to different environmental conditions or at different release rates that are more in line with plant uptake systems. And so, as well as that, here's some examples of some of the materials so far that we've been testing, different combinations of different biodegradable plastics with urea. Here is an example of different minerals with different starches and urea, and we're at the stage at the moment where we're looking at the release rate of these things in water as well as in sand soil systems with different microbial interactions to see how quickly they're going to be released as compared to traditional urea or as compared to other coated or currently available commercial products. And then also there is potential there for there to be some gel or encapsulations of microbial products that also can be incorporated into this material also. Just to focus more on, again, we've been talking a lot about agricultural waste, but generally across the population there is a move towards more recycling and closing of nutrient loops. And in urban settings, currently there's some pilot plants looking at how urine recycling is becoming really a reality. So currently in Brisbane there is a toilet block that is got this setup where they are extracting urine and using this U-Gold technology, which is a collaboration between University of Queensland and the urban utilities in being able to extract the nitrogen and phosphorus from this system. And so currently there's equivalent systems that are present in Europe, and they're producing this product here called Oran, which is marketed and available for sale as a fertilizer. However, the U-Gold processing technology is an energy neutral system, so they're able to extract these elements at the pilot plant. And so we're currently working with the engineers coming up and looking at the best combination of other nutrients and formulations that can be added to the U-Gold product to make it a product that's suitable to grow plants. So moving from urban settings also into agricultural systems where there's a lot of waste streams from agricultural residue processing, different livestock processing. This is an area in terms of nutrient recycling that is currently here and is getting ready to be rolled out in a real way, in a quite a scalable way across different communities. However, really the question that we all need to be thinking about and discussing together is what's the way forward for the circular nutrient economy, and particularly thinking about it in your own setting and cropping system. So how can the nutrients that are currently there, can they be recycled in a better way? What's the safest way to do that, the most efficient way to do that? What are the nutrients that are available in the wastes? How consistent is this nutrient stream going to be? And importantly, is this going to be economically viable? So are we able to extract the nutrients and transport them in an economical way? So at the moment, a lot of the waste and residue is very heavy and has a lot of water, and you have to be able to get rid of the water and be able to transport it to the area that's needed. So whether that is economically doable via transport or if a more localised setting is what's required are questions that we need to answer and resolve, and with an integrated approach, practitioners and researchers, government is required. So here we've been talking about mostly recycling, and there's been a lot of focus on the sugar industry in terms of nitrogen leakage, nitrogen loss because of the close proximity to Great Barrier Reef and the focus on dissolved inorganic nitrogen. But currently there's a really global push to look at sustainable, healthy food production. And recently this article came out in The Lancet, looking at this from a global perspective across all cropping systems in the production of food, where there's a real focus on closing the nutrient loops and being able to use nitrogen and phosphorus more efficiently. And the avenues towards that one is one that you're all very aware of and have been practicing for a long time in applying the right nutrient in the right quantity at the right place at the right time. But also I think the next phase moving forward is the second tier here, where we're looking at how do we recover nutrients efficiently and use them well from the waste streams that we currently have in both the urban and agricultural setting. So I think this is what we really need to be pushing forward to be able to close the nutrient and energy loops and reduce the environmental effects. So this one, we will do this through collaborations with the growers, scientists, the industry push forward, as well as the natural resource management boards within the framework of the Australian government. And I think today and tomorrow is an excellent opportunity for us to start having this discussion and see where we all think that gains can be made or where focus should be put. And hopefully we can all move towards a much more highly efficient tropical agricultural system. And I'd just like to say thanks and acknowledge all the many collaborators that I have, particularly Suzanne Schmidt and Richard Bracken, with whom we've worked very closely in all these projects over the last 15 or so years, and the team here at Plant Nutrient Team at UQ, as well as the chemical engineers, and then as well as at our industry and government partners. Thanks very much.
Well done. Thanks, Nicole. So, yeah, your passion, dedication is really, really inspiring. Nicole's actually going to be in the breakout session after lunch. So if you have some questions for Nicole, we'll utilise that time to be able to answer any of the questions that you might have. Great, so 2019 is the 11th year of Project Catalyst, and it's grown from 20 growers in 2009 to more than 100 active participants today. Project Catalyst is a partnership between the Australian Government, Coca-Cola Foundation, WWF Australia, Bayer Crop Science, and the NRM bodies Reef Catchments Limited, and NQ Dry Tropics and Terrain. A couple of things that I just want to remind you, if you need any refreshments, there is some at the back of the room on both sides, and there's also a fridge just next to the registration desk, compliments of Coca-Cola, so just help yourself at any time. And we're also live streaming our sessions via YouTube. So if you know people that weren't able to be here, you can let them know to catch up with any of the sessions, but it also just means we need to be mindful near the cameras and the microphones if you are in that vicinity. So thanks to Jack from Flow Motion Media for providing that. It's great. Okay, so next I would like to welcome Kim Clayton for the virtual farm tour. So following a 3-day trip to the wet tropics in November of 2018, Kim and Jack were given a personal tour of progressive farms, adopting and trialling biological solutions to reduce the use of inorganic fertilizers and create more life in their soils. So to present that virtual farm tour, please welcome Kim Clayton.
Hello, it's so good to be back with you guys again this year. It really is like a family. And I noticed last night, for those who were here for the welcome function just how much like a family it is, because you were so pleased to see each other. You were all talking and enjoying the evening, so welcome again and thank you very much for being involved. This is one of the biggest Cairns events that we've had, so it's definitely thanks to you and making the effort to be here. As Sally mentioned, you know, we've been touring around in the last couple of years to each of the regions who are hosting this fantastic event to collect stories from the growers. And one of the comments that was made to me by the Coke representatives last year was that it really gave them an understanding of who these people are. Having not set foot on a farm, Sarah, who has attended quite a few forums, was given an insight, I suppose, into exactly why you're doing these things. It's not about the trial and the data necessarily. It's about who you are, why you're doing it, and your story. So Jack and I, once again, had the pleasure of getting on the road, and 3 days we spent driving from Mackay through the wet tropics and visiting farms from Ingham right through to Cairns. We had a fabulous time, met some amazing people and saw some really wonderful rainforests on the way through. So taking that time to get to know the region as well as the people who live and work in it. This video is actually about improving soil health. The biological stimulant one will be a little bit later. So we focused this event around soil, water and the reef because it's imperative that those things work very well together. And having a basic understanding, being a hobby farmer myself and hoping to turn that into a sustainable business in my future, I've been blessed by connection to people like Dr. Nicole Robinson and Suzanne Schmidt. I've attended some of the soil symposiums organised by Simon Matson, who is also a very inspiring person and has shared all of his knowledge from his Nuffield trips worldwide and even inspired me to know more about my soil and how to support it so that the plants growing out of there, whether they be fodder for my goats or the fruits and vegetables that we harvest to eat, are as healthy as they can be and as complete in the nutrition that they require to deliver to your body. So with that in mind, I'd like to now hand over to the farmers we met on our travels, and there's quite a few of them in the room that haven't seen this, so it's the first time they'll be viewing it as well. This is the Soil Health Virtual Farm Tour we filmed in the wet tropics.
For Renzo de Bella, farming has changed significantly since 1928 when his father bought property in Ingham with his two brothers. Mechanisation improved working conditions, but has also contributed to... For Renzo de Bella, farming has changed significantly since 1928 when his father bought property in Ingham with his two brothers. Mechanisation improved working conditions, but has also contributed to a loss of soil condition.
I have acquired this farm here and we bought as a family 2 other farms up the road. It's been a hard trauma for us in the industry because there have been many changes. You take from the manual cane cutter to the loader, the grab loader to load the cane. You have had changes in different, every 10 years there seems to be changes. To save money, we have changed our farming practices from a lot of manual hard work into mechanical harvesting, and we've got machines cutting our cane, preparing our soils. I'm happy to see my grandson behind you being the part of the 4th generation, and he's a worker and works like mad. He can't stop him, hey, smiling, smiling, look at him.
And then I've always kind of like plants and trying new machines and see which stuff, how it works in the paddock and stuff like that.
He's shown an interest in the land, which is a very vital thing in Australia. And I'm proud to see what he does in the field. He can't only live on sugarcane, that he's looking at new innovations of planting other crops like ssoybeans, sweet potatoes, watermelon, pumpkins and that, to try and supplement the earnings that he gets off the farm.
Why don't we have other crops? So if one price is low, surely one's a bit better than the other. At least you've got a variety of crops to choose from and different sources of income harvesting at different times. I used my money that I was going to buy a ute with, and I ended up putting it towards some sweet potato gear. So I got a harvester that I'll be using to pull along, or put it on the back of one of the tractors, a harvester to tow along with a side elevator that I'll put behind. I'll sideload the potatoes in, come into this shed down that bay of the shed. They come through a crate tipper into a washer and dryer out on the other end, just a turntable dryer. Normally disc the paddock once or twice, depending on the soil type, and we pull this bed former straight in. And this year, because it's dry, we haven't planted our beans yet, but as soon as we get moisture, we'll go back in with this bed former again. We've just got the front disc on the front, we can actually go back into existing beds, and then we'll plant the beans.
A passionate researcher and soil health advocate, Hayden's father Lawrence has been inspirational.
We've been yielding up to 30 tonnes per hectare biomass crops in our fallow crops. We're running mixed fallows, so we're mixing soybean, cowpea, lab, sunflowers and a number of other. And at the moment we've got some trial work through Project Catalyst to actually assess which species do what.
We used to work our crops at least 12-15 times each block. Today we harvest green for the specific reason of leaving that trash as a moisture retention and fertilizer, because the trash will rot in the wet season. And we are trying to stop compaction.
I would like to do one day, buy an extra tractor and do a one-pass tillage system, get a tractor with front linkage and place a mulcher on the front to mulch the beans down, followed by a bed former, so you can put beds and mulch at the same time, and we only have to do one wavy disc cultivation pass just before the build planter arrives. If you over-till, they'll come dry and they've got no life in them. To make it a healthy soil, you don't want it too fine like it's just like cement.
Hayden has learnt that diversifying into other crops requires capital investment and planning.
Well, we need irrigation. We have no irrigation for cane, so we have to get a pump, water licenses or allocations that we've had trouble getting. Learning about T-tape irrigation and laying plastic and different chemicals and herbicides to spray on watermelons, pumpkins and sweet potatoes. I only did one row of watermelons, so I think we'll probably get 3 more tonne out of that one row still, because there's heaps of watermelons in there, but I've already picked half a tonne. Last year when we had our mixed crop just over there, the cane grew. We planted the cane in June. The cane grew that fast, by Christmas it was already lodged and fully over. We've got to harvest it at Christmas time if we wanted to.
The whole thing is that you've got to try and educate the farmer to changes. I have found it hard to adapt to new farming situations, and if we don't look at changes, it's the people that you've got to change in their heads. They think that manna comes from heaven. You've got to make things happen to be able to produce something.
Well, Nana always tells me he's proud of me, and always inspires me to try new things, even though he did it differently, and try different farming techniques to improve our productivity.
Currently trialling mixed species cover crops, Ray Zamora follows in his father's progressive footsteps as an early adopter of anything that improves outcomes for the environment.
We've got to keep it, if not better, for our next generations to come. I'd hate for me to be in the ground one day and someone's talking and say, gee, Dad messed that farm up. Like, yeah, no, we've got to regenerate it, and yeah. And when you do that, it's all more profitable, and I don't see the girls are much interested in the farm, but my young fella's shown a bit of sign there. So yeah, hopefully one day he might like to run the farm, and yeah, that'd be great to work with your son.
As farmers continue to embrace technology, Ray admits that might be something his son Rory will manage.
I'm a bit behind the ball in that game. I use the GPS for guidance mainly, with the controlled traffic and everything, and I just do manually recording in a diary. You know, like even the GPS, they're great when they're working, but I've spent half a day in the paddock getting nothing done because it won't work, and yeah, it scrambles my brain sometimes.
A regular on social media, not all technology confounds Ray. He's also mastered online research.
Oh, I've learnt absolutely heaps. Just going on Google, watching YouTube clips on professional people. Yeah, you learn about, you know, lactobacillus, bacillus subtilis, all these different bugs and mycorrhizae fungi. There's just a whole new world out there that us growers have never been taught before. And it's good to see the industry now is getting some traction in that area, and yeah, we're making some good moves, I think. It changes your mindset when you do a Regen Ag course. The object of the day normally for a farmer is, what can I kill? Well, we're starting to think a bit different now, like what can we make grow? In the crops I've been using tillage radish, sun hemp, soys, Japanese millet, yeah, cowpeas, sorghum Sudan grass, and yeah, like a radish, it'll be scavenging excess nitrogen that's in the soil. Other plants bring up phosphorus, you know, yeah, other plants bring up zinc. You know, all plants have got a different role to play in nature. Carcass of a radish, and we call that a nutrient storage vessel. It's sucked up all those nutrients, and I can't break it, but it's going to release all that back into the soil. And normally, like if you've got weeds in your paddock, that's a sign of lacking in nutrition, and they're trying to restore that ground by using the root exudates, you know, putting the liquid carbon back into the ground, and eventually that ground gets so good that the weeds won't want to grow there.
Following growers like Ray Zamora and his mixed species trials, Adrian Darvaneza is hoping to adopt some of the methods in future to improve his own soils.
Yeah, I've seen a few of what some of the other guys are doing down at Ingham and stuff like that, and quite probably not there yet. You know, what they're doing is inspirational, but it's each area I think has got their own challenges. Like where I am, I've got a lot of hills and stuff like that, so I'm sort of limited to what I can and can't do. I can't work my ground up in December, because if I do, it'll just all wash away, you know, and it'll end up in the creek and then go out to the reef, and that's defeating the purpose. So at the moment, I'm just trying to get my head around what I'm doing at the moment myself, if that makes sense. Yeah. Productivity is probably the driving force, I guess, but the way I can become productive is through better soils, better management of time, along those sorts of lines. So the way I was thinking was, if I could get my planting done in May, that would give me more time to do other farm work through the season. And with the zero-till planting, I know that I'm getting less erosion. That was one of the fears I had about planting early, was that you can get a heavy downpour of rain and it can cause bad erosion. But what I saw this year after I planted was pretty wet. Not heavy rain, but it rained every day for weeks and weeks. So, and I got no erosion.
Heavy rain has been the catalyst for change adoption across entire farming systems along the Queensland coast.
As far as runoff, well, for a start, you've got to go to the 1.8 or 1.9 metre system, so that you've got less uncompacted ground. And when you've got less uncompacted ground, you've got more water going through the ground rather than running off. And when you've got better soil health, it's got a chance to hold onto that nutrient. If it's got, you know, a lot of fungi and stuff in the ground, it can filter it on the way through.
Through my father's time, he's done a lot of innovation himself, you know, like not just converting from horses to tractors, but he's also done a lot of work zonally working the ground in the past. So with the zero-till planting, it's something that I've sort of carried that legacy on, as trying to do a bit of innovation, you know, trying to And when Dad did the zonal till rotary arcing, the neighbours started doing it too, you know, so they saw the advantage of that, and so hopefully now people see what I'm doing, and if it works, they might consider doing it as well.
Yeah, I'll put it down to a holistic approach. There's no one silver bullet. Yeah, it's a whole farming system, starting with your fallow, your controlled traffic, your nutrients.
Yeah, number one, productivity. Number two, soil health, I guess. And three, all these things contribute to being a better stewardship to the environment, I suppose. So, you know, we can't ignore the reef. We can't just say we're farmers and they're fishermen. You know, we're all living in this one community together.
Yeah, well, on the farm here we've got a series of wetlands that the water travels through. We keep all our headlands slashed. There's jabberoos, crocodiles of course, and there's plenty of fish. We go down there fishing all the time. Yeah, and that's part of the ecosystem too. You know, it's not all about nitrogen running off to the reef. It's about a whole holistic approach. Yeah, as far as nutrient and pesticides, and yeah, we've had school kids out here this year doing a geography thing, and with Fiona George with the terrain mob, and they took water samples and the nitrate levels come back at 0, so that's a win-win. I mean, it was no good for them because they wanted to actually see something, but that's what the results said, that there was 0 in the water.
Sugar at the moment is getting a bad rap, you know, because it's bad for you, it's the toxic thing and this and that, but I often joke that everyone hates sugar, but everyone likes to eat cake. Which is true.